digestion in the oral cavity, stomach. The primary functions of the digestive system is to transfer nutrients, water, and electrolytes from the food we eat into the body's internal environment. Ingested food is essential as an energy source or fuel from which the cells can generate ATP to carry out their particular energy-dependent activities, such as active transport, contraction, synthesis, and secretion. Food is also a source of building supplies for the renewal and addition of body tissues. The act of eating doesn't automatically make the preformed organic molecules in food available to the body cells as a source of fuel or building blocks. The blood first must be digested or biochemically broken down into small, simple molecules that can be absorbed from the digestive tract into the circulatory system for distribution to the cells. Normally, about 95% of the ingested food is made available for the body's use. Thus, the sequence in nutrient acquisition is ingestion, digestion, absorption, distribution, and usage. The digestive tract and accessory digestive organs make up the digestive system. The digestive system consists of the digestive gastrointestinal tract plus the accessory digestive organs. The accessory digestive organs include the salivary glands, the exocrine pancreas, and the biliary system, which is composed of the liver and gallbladder. These exocrine organs lie outside the digestive tract and empty their secretions through duct into the digestive tract lumen. The digestive tract is essentially a tube about 4.5 meters in length in its normal contractile state. Running through the middle of the body, the digestive tract including the following organs: mouth, pharynx, esophagus, stomach, small intestine consists of the duodenum, jejunum, and ileum, large intestine, the cecum, appendix, colon, and rectum, and anus. Also, these organs are continuous with one another. They are considered as separate entities because of their regional modifications, which allow them to specialize in particular digestive activities. Because the digestive tract is continuous from the mouth to the anus, the lumen of this tube, like the lumen of straw, is continuous with the external environment. As a result, the content within the lumen of the digestive tract are technically outside the body. Just the soda you suck through straw is not part of the straw. Only after substances has been absorbed from the lumen across the digestive tract, wall is the considered part of the body. This is important because conditions essential to the digestive process can be tolerated in the digestive tract lumen that could not be tolerated in the body proper. Consider the following examples. The pH of the stomach contents fall as low as 2 as a result of the gastric secretion of the hydrochloric acid. Yet in the body fluids, the range of pH compatible with life is 6.8 to 8.0. The digestive enzymes that hydrolyze the protein in food would also destroy the body's own tissues that produce them. Protein is the main structural component of cells. Therefore, once these enzymes are synthesized in inactive form, they are not activated until they reach the lumen, where they actually attack the food outside the body, that is, within the lumen, thereby protecting the body tissues against self-digestion. In the lower part of the intestine exist quadrillions of living microorganisms that are normally harmless and even beneficial. Yet, if these same microorganisms enter the body proper, as may happen with the ruptured appendix, then may be extremely harmful or even lethal. Foodstuffs are complex foreign particles that would be attacked by the immune system if they were in contact with the body proper. However, the foodstuffs are digested within the lumen into absorbable units such as glucose, amino acids, and fatty acids that are indistinguishable from these simple energy-rich molecules already present in the body. The digestive system performs four basic digestive processes: motility, secretion, digestion, and absorption. The term motility refers to the muscular contractions that mix and move forward the contents of the digestive tract. Also, the smooth muscle in the walls of the digestive tract is phasic smooth muscle that displays action potential induced bursts of contraction. It also maintains a constant low level of contractions known as tone. Tone is important in maintaining steady pressure on the contents of the digestive tract, as well as in preventing its walls from remaining permanently strained following digestion. Two basic types of phasic digestive motility are superimposed on this ongoing tonic activity: propulsive movements and mixing movements. Propulsive movements propel or push the contents forward through the digestive tract, with the rate of propulsion varying depending on the functions accomplished by the different regions. That is, the content are moved forward in given segment and appropriate velocity to allow that segment to do its job. For example, transit of food through the esophagus is rapid, which is appropriate because this structure merely serves as a passageway from the mouth to the stomach. In comparison, in the small intestine, the main site of digestion and absorption, the contents are moved forward slowly, allowing time for the breakdown and absorption of food. Mixing movements serve a twofold functions. First, by mixing food with the digestive juices. These movements promote digestion of the food. Second, they facilitate absorption by exposing all parts of the intestinal contents to absorption surface of the digestive tract. Contraction of the smooth muscle within the walls of the digestive organs accomplishes movement of material through most of the digestive tract. The exception are the ends of the tract, the mouth, through the early part of the esophagus, at the beginning and the external anal sphincter, at the end where motility involves skeletal muscles rather than smooth muscle activity. Accordingly, the acts of chewing, swallowing, and defecation have voluntary components, because skeletal muscle is under voluntary control. By contrast, motility accomplished by smooth muscle throughout the rest of the tract is controlled by complex involuntary mechanisms. number of digestive juices are secreted into the digestive tract lumen by exocrine glands along the road, each with its own specific secretory product. Each digestive secretion consists of water, electrolyte, and specific organic constituents important in digestive process such as enzyme, bile salts, or mucus. The secretory salts extract from the plasma large volumes of water and the raw materials necessary to produce their particular secretion. Secretion of all digestive juices requires energy, both for active transport of some of the raw materials into the cells and for synthesis of secretory products by the endoplasmic reticulum. On appropriate neural or hormonal stimulation, the secretions are released into the digestive tract lumen. Normally, the digestive secretions are reabsorbed in one form or another back into the blood after the participation in digestion. Failure to do so results in loss of these fluids that has been borrowed from the plasma. Furthermore, endocrine cells located in the digestive tract wall secrete gastrointestinal hormones into the blood that help control digestive motility and exocrine gland secretion. Humans consume three different biochemical categories of energy-rich foodstuffs: carbohydrates, proteins, and fats. These large molecules cannot cross plasma membranes intact to be absorbed from the lumen of the digestive tract into the blood or lymph. The term digestion refers to the biochemical breakdown of the structurally complex foodstuff of the diet into smaller absorbed units by the enzyme produced with the digestive system as follows. First, the simplest carbohydrates are the simplest sugars or monosaccharides such as glucose, fructose, and galactose, very few of which are normally found in the diet. Most ingested carbohydrate is in the form of polysaccharides, which consist of chain of interconnected glucose molecules. The most common polysaccharides consumed in starch derived from plant sources. In addition, meat contains glycogen, the polysaccharide storage from the glucose in muscle. Cellulose, another dietary polysaccharide found in the plant walls, cannot be digested into its constituents, monosaccharides, by the digestive juices humans secrete. Thus, it represents the indigestible fiber or bulk of our diets. Besides polysaccharides, a lesser source of dietary carbohydrate is in form of disaccharides, two sugar molecules, including sucrose, table sugar, which consists of one glucose and one fructose molecule, and lactose, milk sugar, made up of one glucose and one galactose molecule. Through the process of digestion, starch, glycogen, and disaccharides are converted into their constituents, monosaccharides, principally glucose with small amount of fructose and galactose. These monosaccharides are the absorbable units for carbohydrates. Second one, dietary protein consists of various combinations of amino acids held together by peptide bonds. Through the process of digestion, proteins are degraded primarily into their constituent amino acids as well as few small polypeptides, both of which are the absorbable unit for protein. Third one, Most dietary fats are in form of triglycerides, which are neutral fats. Each consists of glycerol with three fatty acid molecules attached. During digestion, two of the fatty acid molecules are split off, leaving a monoglyceride, a glycerol molecule, with one fatty acid molecule attached. Thus, the end products of fat digestion are monoglycerides and free fatty acids, which are absorbable unit of fat. Digestion is accomplished by enzymatic hydrolysis. By adding water at the bond site, enzymes in the digestive secretion break down the bonds that hold the small molecular subunit within the nutrient molecules together, thus setting the small molecule free. The removal of water at the bond site originally joined the small subunits to form nutrient molecules. Hydrolysis replaced the water and frees the small absorbable units. Digestive enzymes are specific in the bonds they can hydrolyze. As food moves through the digestive tract, it's subjected to various enzymes, each of which breaks down the food molecules even further. In this way, large food molecules are converted to simple absorbable units in a progressive, stepwise fashion, like assembly line in reverse as the digestive tract content are propelled forward. In the small intestine, digestion is completed and most absorption occurs. Through the process of absorption, the small absorbable units that result from digestion, along with water, vitamins, and electrolytes, are transferred from digestive tract lumen into the blood or lymph. The oral cavity is the entrance to the digestive tract. Entry to the digestive tract is through the mouth or oral cavity. The opening is formed by the muscular lips, which help produce, quiet, and contain the food in the mouth. The lips also serve non-digestive functions. They are important in speech. Articulation of many sounds depend on the particular lip formation, and as sensory receptors in interpersonal relationship. The lips are endowed with especially well-developed tactile touch sensation. The palate, which forms the arched roof of the oral cavity, separates the mouth from the nasal passages. Its presence allows breathing and chewing or sucking to take place simultaneously. Handing down from palate in the rear of the throat is a ganglion projection, the uvula, which plays important role in sealing of the nasal passages during swallowing. The tongue, which forms the floor of the oral cavity, is composed of voluntary control skeletal muscle. Movements of the tongue are important in guiding food within the mouth during chewing and swallowing, and also play important role in speech. Furthermore, the major taste buds are located on the tongue. Taste receptor cells are located primarily within tongue. The chemoreceptors for taste sensation are packaged in taste buds, about 10,000 of which are present in the oral cavity and throat, with the greatest percentage of the upper surface of the tongue. A taste bud consists of about 50 long, spindle-shaped taste receptors cell packaged with supporting cells in arrangement like slices of an orange. Each test bud has a small opening, the taste pore, through which fluid in the mouth come into contact with the surface of its receptor cells. Taste receptor cells are modified epithelial cells with many surface folds, or microvilli, that protrude slightly through the taste pore, greatly increasing the surface area exposed to the oral contents. The plasma membrane of the microvilli contains receptor sites that bind selectively with chemical molecules in the environment. Only chemicals in solution, either ingested liquids or solids that have been dissolved in saliva, can attach to receptor cells and evoke the sensation of taste. Binding of taste-provoking chemicals, testing with receptor cells alters the cell's ionic channels to produce depolarization receptor potential. Like the other special sense receptors, depolarizing receptor potential opens voltage gate calcium channels, leading to the entry of the calcium, which promotes release of neurotransmitter. This neurotransmitter in turn initiated action potentials within terminal ending of afferent nerve fibers with which the receptor cell synapses. Most receptors are carefully sheltered from direct exposure to the environment, but the taste receptor cells, by virtue of the task, frequently come into contact with potent chemicals, unlike the eye or eye receptors, which are irreplaceable. Taste receptors have a lifespan of about 10 days. Epithelial cells surrounding the taste but differentiate first into supporting cell and the interoceptor cells to constantly renew the taste bud components. Terminal afferent endings of several cranial nerve synapses with taste buds in various regions of the mouth. Signals in the sensory inputs are conveyed via synaptic stops in the brainstem and thalamus to the cortical gustatory area, a region in the parietal lobe adjacent to the tongue area of the somatosensory cortex. Unlike most sensory input, the gustatory pathway are primarily uncrossed. The brainstem also projects fibers to the hypothalamus and limbic system to add affective dimensions, such as whether the taste is pleasant or unpleasant, and to process behavioral aspects associated with the taste and smell. Taste discrimination is coded by pattern of activity in various taste bud receptors. We can discriminate among thousands of different taste sensations, yet all tastes are varying combinations of 5 primarily tastes: salty, sour, bitter, and umami. Umami, a meaty or savory taste, has recently been added to the list of primarily tastes. Another new taste sensation has also been proposed: fatty. Scientists have identified a potential sensor in the mouth for long-chain fatty acids. Each receptor cell responds in varying degrees of all 5, maybe 6 primarily taste buds generally responds preferentially to one of the taste modalities. 5 taste discrimination beyond the primary taste depends on suitable differences in the stimulation pattern of all the taste buds in response to various substances, similar to the variable stimulation of the 3 cone types that gives rise to the range of color sensations. Receptor cells use different pathways to bring about depolarizing receptor potential in response to each tasting category. Salty taste is stimulated by chemical salts, especially table salt, directly entry of positively charged sodium ions through specialized sodium channels in the receptor cell membrane, a movement that reduces the cell internal negativity is responsible for receptor depolarization in response to salt. Sour taste is caused by acids which contain a free hydrogen ion. The citric acid content of lemons, for example, accounts for the distinctly sour taste. Depolarization of the receptor cell by sour tasting occurs because hydrogen block potassium channels in the receptor cell membrane. The result decrease in the passive movement of positively charged potassium ions out of the cell reduces the internal negativity, producing depolarizing receptor potential. Sweet taste is evoked by particular configuration of glucose. From evolutionary perspective, we crave sweet foods because they supply necessary calories in readily usable form. However, other organic molecules with similar structure but no calories, such as saccharin, aspartame, sucralose, and other artificial sweeteners, can also interact with sweet receptors binding sites. Binding of glucose or another chemicals with the taste cell receptors activate G protein, which turns on the amino monophosphate, second messenger pathway in the taste cell. The second messenger pathway ultimately results in phosphorylation and blockage of potassium channels in the receptor cell membrane, leading to depolarizing receptor potential. Bitter taste is elicited by a more chemical diverse group of tasting than the other taste sensation. For example, alkaloids as well as poisonous substances all taste bitter, presumably as a protective mechanism to discourage ingestion of these potentially dangerous compounds. Taste cells that detect bitter flavors process 50 to 100 bitter receptors, each of which responds to different bitter flavor. Because each receptor cell has this diverse family of bitter receptors, a wide variety of unrelated chemicals all taste bitter despite the diverse structures. This mechanism expands the ability of the taste receptor to detect a wide range of potentially harmful chemicals. Umami taste, which was first identified and named by Japanese researchers, is triggered by amino acids, especially glutamate. The presence of amino acids is found in meat, for example, serves as marker for desirable, nutritionally protein-rich food. Glutamate binds to a G proteins coupled receptors and activates second messenger pathway. In addition to giving us our sense of meaty flavors, this pathway is responsible for the digestive taste of all flavor additive monosodium glutamate, which is especially popular in Asian dishes. In addition to input from the taste receptors, taste perception is also influenced by information derived from other receptors, especially odor. When you temporarily lose your sense of smell because of swollen nasal passageway during cold, your sense of taste is also markedly reduced, even though your taste receptors are unaffected by the cold. Other factors affecting taste include temperature and texture of the foods, as well as physiological factors associated with past experiences with food. How the digestive cortex accomplished in the complex perceptual processing of taste sensation is currently not known. teeth are responsible for chewing. The first step in the digestive process is mastication or chewing, the motility of the mouth that involves the slicing, tearing, grinding, and mixing of ingested food by the teeth. The teeth are firmly embedded in and protrude from the jawbones. The exposed part of tooth is covered by enamel, the hardest structure of the body. Enamel forms before the tooth eruption by special cells that are lost in the tooth erupts. Because enamel cannot be regenerated after the tooth has erupted, any defects that develop in enamel must be patched by artificial links, or else the surface will continue to erode into the underlying living pulp. The upper and lower teeth normally fit together when the jaw is closed. This occlusion allows food to be ground and crushed between the two surfaces. When the teeth don't make proper contact with one another, they cannot accomplish their normal cutting and grinding action adequately. Such malocclusion results for abnormal positioning of the teeth and is often caused either by overcrowding of teeth too large for the available jaw space or by one jaw being displaced in relation to the other. In addition to ineffective chewing, malocclusion can cause abnormal wearing of affected tooth surfaces and dysfunction and pain of temporomandibular joint, where the jaws articulate with each other. Malocclusions can often be corrected by applying braces, which exert prolonged, gentle pressure against the teeth to move them gradually to the desired position. The teeth can exert forces much greater than those necessary to eat ordinary food. For example, the molares in adult can exert crushing force of up to 200 pounds, which is sufficient to crack a hard nut, but ordinarily these powerful forces are not used. In fact, the degree of occlusion is more important than the force of the bite in determining the efficiency of chewing. The functions of chewing are to grind and break food up into smaller pieces, to facilitate swallowing, and to increase the food surface area on which salivary enzymes will act. Second, to mix food with saliva. And third, to stimulate the taste buds. The latter not only gives rise to the pleasurable subjective sensation of taste, but also, in feed-forward fashion, reflexly increases salivary, gastric, pancreatic, and bile secretion to prepare for the arrival of food. The act of chewing can be voluntary, but most chewing during a meal is a rhythmic reflex brought about by activation of the skeletal muscles of the jaws, lips, cheeks, and tongue in response to the pressure of food against the oral tissues. Saliva begins carbohydrate digestion, is important in oral hygiene, and facilitates speech. Saliva, the secretion associated with the mouth, is produced largely by three major parts of salivary glands that lie outside the oral cavity and discharge saliva through short ducts into the mouth. Saliva is about 99.5% water and 0.5% electrolytes and protein. The salivary salt concentration is only 1/7 of that in the plasma, which is important in perceiving salty taste. Similarly, discrimination of sweet taste is enhanced by the absence of glucose in the saliva. The most important salivary proteins are amylase, mucus, and lysozyme. They contribute to the functions of saliva as follows. First, saliva begins digestion of carbohydrate in the mouth through action of salivary amylase, an enzyme that breaks polysaccharides down into maltose disaccharides consisting of 2 glucose molecules. Second, saliva facilitates swallowing by moisturizing food particles, thereby holding them together and by providing lubrication through the presence of mucus, which is thick and slippery. Third, saliva exerts some antibacterial action by fourfold effect. First, by lysozyme, an enzyme that lyses or destroys certain bacteria by breaking down their cell walls. Second, by a binding glycoprotein that binds antiglobulin A antibodies. Third, by lactoferrin, which tightly binds to iron needed for bacterial multiplication. And fourth, by rinding away material that may serve as food source for bacteria. Fourth, saliva serves as a solvent for molecules that stimulate the taste buds. Only molecules in solutions can react with taste bud receptors. You can demonstrate this for yourself. Dry your tongue and then drop some sugar on it, and you cannot taste the sugar until it's moistured. The flow of saliva also flushes away the food particles on taste buds so that you can taste the next bite of food. Fifth, saliva aids speech by facilitating movements of the lips and tongue. It's difficult to talk when your mouth feels dry. 6. Saliva plays an important role in oral hygiene by helping keep the mouth and teeth clean. The constant flow of saliva helps flush away food residue, foreign particles, and all epithelial cells that have shed from the oral mucosa. Saliva's contributions in the regard is apparent to anyone who has experienced foretaste in the mouth when salivation is suppressed for a while, such as during fever or states of prolonged anxiety. 7. Saliva is rich in bicarbonate buffers, which neutralize acids in food, as well as acids produced by bacteria in the mouth, thereby helping prevent dental caries. Despite these many functions, saliva is not essential for digesting and absorption foods, because enzymes produced by pancreas and small intestine can complete food digestion even in the absence of salivary and gastric secretion. The main problem associated with diminished salivary secretion, conditions known as xerostomia, are difficulty in chewing and swallowing, inarticulate speech, unless frequent sips of water are taken when talking, and a rampant increase in dental caries unless special precautions are taken. Salivary secretion is continuous and can be reflexly increased. On average, about 1 to 2 liters of saliva are secreted per day, ranging from the continuous spontaneously basal rate of 0.5 ml per minute to a maximum flow rate of about 5 ml per minute, in response to a potent stimulus such as sucking or lemon. The continuous basal secretion of saliva in the absence of apparent stimuli is brought about by constant low-level stimulus by the parasympathetic nerve endings that terminate in the salivary glands. This basal secretion is important in keeping the mouth, the throat, moist at all times. In addition to this continuous low-level secretion, salivary secretion may be increased by two types of salivary reflexes: the simple and conditioned salivary reflexes. Simple and conditioned salivary reflexes. The simple salivary reflexes occur when chemoreceptors and pressure receptors within the oral cavity respond to the presence of food. On activation, these receptors initiate impulses in afferent nerve fibers that carry the information to the salivary center, which is located in the medulla of the brainstem, as are all the brain centers that control digestive activities. The salivary center, in turn, sends impulses via the extrinsic autonomic nerves to the salivary glands to promote increased salivation. Dental procedures promote salivary secretion in the absence of food because these manipulations activate pressure receptors in the mouth. With the conditioned or acquired salivary reflexes, salivation occurs without oral stimulation. Just thinking about seeing, smelling, or hearing the preparation of pleasant food initiates salivation through this reflex. All of us have experienced such mouth-watering in anticipation of something delicious to eat. This reflex is learned response based on previous experience. Input that arise outside the mouth and are mentally associated with pleasure of eating act through the cerebral cortex to stimulate the medullary salivary center. Autonomic influences of salivary secretion. The salivary center controls the degree of salivary output by means of autonomic nerves that supply the salivary glands. Unlike the autonomic nervous system, elsewhere in the body, sympathetic and parasympathetic responses in the salivary glands are not antagonistic. Both sympathetic and parasympathetic stimulation increase salivary secretion, but the quantity, characteristics, and mechanism are different. Parasympathetic stimulation, which exerts the dominant role of salivary secretion, produces a prompt and abundant flow of watery saliva that is rich in enzymes. Sympathetic stimulation, by contrast, produces a much smaller volume of thick saliva that is rich mucus, because sympathetic stimulation elicits a smaller volume of saliva. The mouth feels drier than usual during circumstances when the sympathetic system is dominant, such as stress situation. Thus, people often expressing a dry feeling in the mouth when they are nervous about giving a speech. Salivary secretion is the only digestive secretion entirely under neural control. All other digestive secretions are regulated by both nervous system, reflexes, and hormones. Digestion in the mouth is minimal. No absorption of nutrients occurs. Digestion in the mouth involves the hydrolysis of polysaccharides into disaccharides by amylase. However, most digestion by this enzyme is accomplished in the body of the stomach after the food mass and saliva have been swallowed. Acid inactivates amylase, but in the center of the food mass, where stomach acid has not yet reached, this salivary enzyme continues to function for several more hours. No absorption of foodstuff occurs from the mouth. Importantly, some drugs can be absorbed by the oral mucosa, a prime example being nitroglycerin, a vasodilatator drug sometimes used by cardiac patients to relieve anginal attack associated with myocardial ischemia. The pharynx in the cavity at the rear of the throat. It acts as a common passageway for both the digestive system by serving as a link between the mouth and esophagus for food, and the respiratory system by providing access between the nasal passages and trachea for air. This arrangement necessitates mechanism to guide food and air into the proper passageway between the pharynx. Housed within the side walls of the pharynx are the tonsils, lymphoid tissue that are part of the body's defense team. The motility associated with the pharynx and esophagus is swallowing. Most of us think of swallowing as the limited act of moving food out of the mouth into the esophagus. However, swallowing actually is the entire process of moving food from the mouth through the esophagus into the stomach. Swallowing is sequentially programmed, all or none reflex. Swallowing is initiated when a bolus or ball of chewing or liquid food is voluntarily forced by the tongue to the rear of the mouth into the pharynx. The pressure of the bolus stimulates pharyngeal pressure receptors, which send afferent impulses to the swallowing center located in the middle of the brainstem. The swallowing center then reflexly activates in the appropriate sequence the muscles involved in swallowing. Swallowing is the most complex reflex in the body. Multiple highly coordinated responses are triggered in specific all or none pattern over a period of time to accomplish the act of swallowing. Swallowing is initiated voluntarily, but once begun, it cannot be stopped. Perhaps you have experienced this when a large piece of half candy inadvertently slipped to the rear of your throat, triggering an unintentional swallow. During the oropharyngeal stage of swallowing, food is prevented from entering the wrong passageways. Swallowing is divided into the oropharyngeal stage and the esophageal stage. The oropharyngeal stage lasts about 1 second and consists of moving the bolus from the mouth through the pharynx and into the esophagus. When the bolus enters the pharynx, it must be directed into the esophagus and prevented from entering the other openings that communicate with the pharynx. In other words, food must be kept from re-entering the mouth, from entering the nasal passages, and from entering the trachea. All of this managed by the following coordinate activities. The position of the tongue against the hard palate keeps food from re-entering the mouth during swallowing. The uvula is evaluated and lodges against the back of the throat, sealing off the nasal passage from the pharynx so that food does not enter the nose. Food is prevented from entering the trachea primarily by elevation of the larynx and tight closure of the vocal folds across the laryngeal opening or glottis. The first part of the trachea is the larynx or voice box, across which the vocal folds are stretched. During swallowing, the vocal folds serve a purpose unrelated to speech. Contraction of laryngeal muscles allows the vocal folds in the tight apposition to each other, thus sealing the glossy entrance. Also, the bolus tilts a small flap of cartilaginous fissure, the epiglottis, backward down over the closing glottis as further protection from food entering the respiratory airways. The person does not attempt futile respiratory efforts when the respiratory passages are temporarily sealed off during swallowing, because the swallowing center briefly inhibits the nearby respiratory center. With the larynx and trachea sealed off, pharyngeal muscles contract to force the bolus into the esophagus. Food is prevented from entering the trachea primarily by elevation of the larynx and tight closure of the vocal folds across the laryngeal opening or glottis. The first part of the trachea is the larynx or voice box, across which the vocal folds are stretched. During swallowing, the vocal folds serve a purpose unrelated to speech. Contraction of laryngeal muscles aligns the vocal folds in tight apposition to each other, thus sealing the glottis entrance. Also, the bolus tilts a small flap of cartilaginous tissues, that epiglottis backward down over the closed glottis as further protection from food entering the respiratory airways. The person doesn't attempt futile respiratory efforts when the respiratory passages are temporarily sealed off during swallowing, because the swallowing center briefly inhibits the nearby respiratory center. With the larynx and trachea sealed off, pharyngeal muscles contract to force the bolus into the esophagus. The esophagus is a fairly strange muscular tube that extends between the pharynx and stomach. Lying for most part in the thoracic cavity, it penetrates the diaphragm and joins the stomach in the abdominal cavity a few centimeters below the diaphragm. The esophagus is guarded at both ends by sphincters. A sphincter is a ring-like muscular structure that, when closed, prevents passage through the tube it guards. The upper esophageal sphincter is the pharyngoesophageal sphincter, and the lower esophageal sphincter is the gastroesophageal sphincter. Except during swallow, pharyngoesophageal sphincter remains closed as a result of neurally induced contraction of the sphincter's circular skeletal muscle. Tonic contraction of this upper esophageal sphincter keeps the entrance to the esophagus closed to prevent large volumes of air from entering the esophagus and stomach during breathing. Instead, air is directed only into the respiratory airways. Otherwise, the digestive tract would be subjected to large volumes of gas, which would lead to excessive eructation or burping. During swallowing, this sphincter opens and allows the bolus to pass into the esophagus. Once the bolus has entered the esophagus, the pharyngoesophageal sphincter closes, the respiratory airways are opened, and breathing resumes. The oropharyngeal stage is complete, and about 1 second has passed since the swallow was first initiated. Peristaltic waves push food through the esophagus. The esophageal stage of the swallow now begins. The swallowing center triggers a primary peristaltic wave that sweeps from the beginning of the end of the esophagus, forcing the bolus ahead of it through the esophageal to the stomach. The term peristalsis refers to ring-like contraction of the circular smooth muscle that moves progressively forward, pushing the bolus into a relaxed area ahead of contraction. The peristaltic waves take about 5 to 9 seconds to reach the lower end of the esophagus. Progression of the wave is controlled by the swallowing center, with innervation by means of the vagus. If a large of sticky swallowed bolus, such as bite of peanut butter sandwich, fails to be carried along to the stomach by the primarily peristaltic wave, the longer bolus distends the esophagus, stimulating pressure receptors within its walls. As a result, a second more forceful peristaltic wave is initiated, mediated by the intrinsic nerve plexus at the level of the distension. These secondary peristaltic waves don't involve the swallowing center, nor in the person airway of their occurrence. Distension of the esophagus also reflexly increases salivary secretion. The trapped bolus is eventually dislodged and moved forward through the combination of lubrication by the extra swell of saliva and the forceful secondary peristaltic waves. Esophageal peristalsis is so effective you could eat an entire meal while you were upside down, and it would all promptly be pushed to the stomach. The gastroesophageal sphincter prevents reflux of gastric contents. Except during swallowing, the gastroesophageal sphincter, which is smooth muscle, in contrast to the upper esophageal sphincter, stays tonically contracted by means of the myogenic activity to maintain barrier between the stomach and esophagus, reducing the change of reflux of acid gastric content into the esophagus. If gastric content do flow backward despite the sphincter, the activity of this content irritates the esophagus, causing the esophageal discomfort known as heartburn. The heart itself is not involved at all. As the peristaltic wave sweeps down the esophagus, the gastroesophageal sphincter relaxed so that the bolus can pass into the stomach. After the bolus has entered the stomach, the swallow is complete and this lower esophageal sphincter again contracts. Esophageal secretion is entirely protective. Esophageal secretion is entirely mucus. In fact, mucus is secreted throughout the length of the digestive tract by mucus-secreting gland cells in the mucose. By lubricating the passage of food, esophageal mucus lessens the likelihood that the esophagus will be damaged by any sharp edges in the newly entering food. Furthermore, it protects the esophageal wall from acid and enzymes in gastric juices if gastric reflux occurs. The entire transit time in the pharynx and the esophagus average a mere 6 to 10 seconds, too short time for any digestion or absorption in this region.